Thank you very much. It's a real pleasure to be here today with you, and I thank you all for coming out on what's starting to look like quite a blizzardy day. But hopefully we'll make it worth your while. Okay. So we're going to continue the theme of DNA, except this time it's going to be literal, all right? And we're going to talk today about me and my genome, or you and your own genome. So I put this question in the little blurb that goes out with it. You know, will you have your genome printed on your health card? You may not even have your own Manitoba health cards yet, I don't know. But soon you will. Will it be on there? Who says yes? Who says no? Yes. The answer is certainly going to be yes. All right. Does everyone know what a genome is? We should probably start with the... Do you know what a gene is? Okay. So genes are small units of DNA that code for proteins, right? There are 22,000 of these in your genome, in each of your cells. And that's kind of a small functional unit of the genome. The genome is all the blueprint, all 6 billion base pairs. It's located on 23 pairs of chromosomes. Every one of these chromosomes is a molecule, single molecule you can stretch out. It will be this long. So 46 of those molecules somehow get compacted up into every one of your cells in your body. So huge blueprint. It's important. And as you just said, most of you know what the genes are. What we're going to talk about today is technologies that have changed our ability to decode that entire blueprint, all 6 billion base pairs, as opposed to maybe 1,000 or 2 for a gene. And so a lot of what we're going to talk about today is the impact that this technology is going to have on, I'll tell you from my perspective, science, engineering, technology. These are all future careers for you in the room. I think you're all here because you have an interest in that. So hopefully it'll spark some innovative ideas for you. And then we'll talk a little bit about what does it mean. What does it mean not as science or medicine, but what does it mean to you sitting right there, right? So hopefully we'll get to that point. It is not a crazy idea. Some people already have their own genomes carrying around on a little memory stick in their pocket. This is the genome of Craig Ventnor. He's a famous geneticist, one of the main pushers of technology and genetics and sequencing, drove the Genome Project, transformed it in its tenure. And of course, when you have a lot of technology and a lot of power and a lot of money and a lot of innovation and you have your own companies, you can actually go ahead and sequence your own genome. So this is a Nature paper. You can look it up. You can go online and check out his genome. It's on the web. I think soon we'll all have our genomes. It'll be on our health cards or associated with our health records. And so let's learn a little bit more about what that means to medicine and what that means to you personally. So here's a pretty busy picture about the history of genetics and genomics. You know, I blew this up on my computer last night. I can't even read some of the things on here myself. But it is interesting if you're a bit of a history buff. You can download it. It's on the web. Get the PDF version. You can actually read it. But I just wanted to make a few points. Genetics has been around since Gregor Mendel did his pea flower experiment in 1850, whatever it was. And for 100 years, lots of important things happened in terms of genetics, looking at single genes, traits where a gene is the result or causes a phenotype, a change in an animal or a plant or organism. In 1991, this is when the Genome Project began. This was kind of one of the first biology big projects, as big as going to the moon in terms of getting the world together behind it, the funds that went into it. And we had enough technology where this was possible. This was going to be a 20-year, and when people say 20-year, they mean like we hope we're done by 40 years. But nonetheless, the value of knowing this code was universally accepted around the planet, certainly here in Canada, America, and Europe. And so this project starts chugging along. Here's where it started. And something really important to this project alone happened just about 10 years into it. What happened there was a technology change. So DNA sequencing is kind of a chemical reaction. You get a little piece of DNA, and you try to figure out what that code is, or where are the T's, A's, C's, and G's. I did this as a postdoc. I've run so many of these gels. You have to read them hand by hand, typing into your Mac on the side, and hoping it all makes sense at the end. There were no databases to compare what lovely sequence I found. So this was really at the edge of it at that time. But what changed was Ventnor and his group, who were doing this competitively against universities and the public enterprise, invented and helped develop a new technology that really made this fast. This was high throughput sequencing. On these new machines, there were no gels, there were no people counting. You load your samples, it runs out, and you could load 20 at a time, not one at a time. And it's all read out in these beautiful little peaks, color-coded. The computer can figure that out. There's no reading of gels. All these files start flying in. Completely changed it. This was about year 7 or 8 in this 40-year project. It was finished 3 years later. Right? So that's the impact of technology, engineering, on this science that I'm going to talk to you today about. If you go to the end of the slide, and this is why I had to get the PDF, like what is at the end of this? It says, to be continued, ... Right? This was published after the genomes were sequenced, and we started to make some sense of it, okay? So I can tell you what happened in the last 8 years. Another really big technological change. In fact, this is a revolution. This is not sequencing or fast, high throughput sequencing. This is, and I quote, massive parallel sequencing. It is unbelievable, almost a completely new approach to sequencing. It all happens on chips, millions of these little arrays at a time. And in fact, you know, in the old days, you hope you got the gene you're interested in and you try to sequence it, right? And then try to make some sense of it. With this machine, you don't even do that anymore. You just take any DNA, blast it into 1 billion pieces, sequence the whole thing, and put all those pieces together afterwards. This was transforming. This machine can do, I don't even know how many hundreds or thousands of times, but I can give you practical examples. Craig Ventnor did his DNA on the old machines. I think that took 2, 3, 4 years. And I don't know how many machines because he owns companies with hundreds of machines. This machine sits on the desktop. It's about this big, right? It's not whole gymnasiums full of the other machines. It will do an entire genome in less than 2 days. 40-year project last decade, 2-day project this decade, right? This is a technological revolution in DNA sequencing. So what does that mean? It means genomes, the whole code, your whole code, are accessible now. This isn't pie in the sky. And you know, we're just looking at here a graph of the cost per genome. So I think this starts in 2001. That's when the first human genome was done. A genome costs $100 million. Okay? This scale here is a little bit of engineering and math. This says, for you computer technologists in the room, or aspiring to be, the whole progress was made simply by faster computers. So this is the rate at which computers get faster, and you can see sequencing genomes gets faster at the rate of your new Apple computer. This is where this technology changed, and this drops precipitously. So at the end of this slide, which is October 2011, it now was under $10,000 to do a whole genome, right? And you can do that in 2 days, runtime. 100 million to 10,000, okay? And this is where, in the last few years, why you may read in Science or see on CNN, this idea of the $1,000 genome. The economists tell us if when we break that $1,000 mark per genome, it's going to be on your health card. It now makes more sense to get the whole code and always have everything you might ever want to know available to you, okay? So, are we there yet? I didn't think so. I thought this was a couple years away. But while I was Googling around looking for nice slides for this talk, out comes this company online, Autogenetics. And there it is advertised right there. It's not even like in a big banner. Your genome, $698. Yeah, that's my price as a scientist, you see. Unbelievable. So we are there. This is really going to happen. How it's going to happen, we can have some discussion about that, all right? So even in the commercial area of the business of science, we have companies coming, and these companies need branding. I really enjoyed your talk beforehand. And if you think this isn't for you sitting around the room, you are wrong. 23andMe, hugely successful company. This is consumer genomics. This is a company that sits right next to Apple for downloading your music, sits right next to Amazon for buying things and getting them delivered a week later when you forgot to order it, and it's a surprise, right? This is a company where, and here it is, this is the main page, same slick kind of really high-tech advertising. Below it is a, how does this work? 4 steps, 4 pictures, you know, order your kit, click here. Kit comes, spit in a tube. We can probably all handle that. Mail said tube back, get an email. Here's your genome. Here's all the things that we think we know about the different small differences in your genome compared to the person sitting next to you. And here's what those small differences might make to your life and your potential for disease. Unbelievable, really. Could you imagine? I couldn't imagine 5 years ago that this would be true. So genomes, your genome is right here. You can do this at Starbucks after, you can do this at lunch, you can order your kit, and off you go. And if you pay a premium, if you join the club pack, you'll get monthly updates. How about that, eh? It's a subscriber service. So this is real. This is really here, really now. It's not just something that's possible. So let's step back a little bit. We've gone from genes, single trait, retinoblastoma gene, cancer of the eye, to genomes, entire genomes, the whole code, all 22,000 coding genes. Now I can tell you, we've got a name for all of these because we like roadmaps. There are 22,000 genes, there's certainly 22,000 names. We only know about what maybe 2,000 of these genes really do. So there's a whole lot of science and discovery yet to go. But this is happening at the same pace as that sequencing curve that I just showed you. It is changing today the way I do science in my lab. It's changing today the way my colleagues do diagnosis in the clinics. It's changing today the way you will make choices about your own health care down the future. Okay? So Mendelian genetics, single gene, single phenotype, to looking at the entire thing all at once. Right? Hugely different way of approaching it. This, if you continue down this straight genetics approach, you know, this is like disease genes. Oh, we found a new disease gene for schizophrenia, right? And we know what to do with that afterwards. The same kind of search goes on for biomarkers. So biomarker is a change that also goes with a disease, but you don't really know whether it causes the disease. You just know that it says this cell or this tissue has this problem. This is kind of our stochastic approach right now. Genomics, this opens up the holiest of holy grails in medicine, complex diseases. Heart disease, cancer, these aren't single gene diseases. These are diseases that have 20, 30, 40 different genes and just the right flavor of them interacting to maybe cause the disease. Certainly, it's shown now that they are causative of it or associated with the cause. But how you put 3 together is a challenge. How you put 20 together is really impossible. This is now tractable. If you can do a whole genome, you get entire information. And so this is what we're going to end up having, hopefully, a better appreciation of. What is personalized medicine? You hear this all the time. Is this about me and my own genome? We'll find out. So, how is genome-wide sequencing changing discovery, diagnosis, treatment? I'm going to show you some science slides, so be prepared. And then we'll get into this question of what is personalized medicine. These are things we do right here. You heard a little bit about what I do. We try to model these mutations or changes in mice, and so we can find out, does it really cause the disease? And if it does, great. We now have a preclinical model to look for therapies. That's what I do. But this idea of using this new technology to find new disease genes is something that is completely transformed. Nature Genetics is the premier journal. It's a nature journal. Started about 15 years ago. Almost every article is about a new gene, a new disease gene, all right? It wasn't that long ago, maybe 6 years ago, it was thought, we're done. There are no more single genes. We're not going to find the rest. We should maybe even close down this journal. Completely changed now. Because Terry Zielinski, a professor here at the university, went after a disease that's probably got a funny name, JRA. There are what we call orphan diseases. So I talked about complex diseases, heart disease, stroke, cancer. These are diseases that, those three alone, probably affect 90% of the kinds of diseases we treat and hope never to get in our lifetime. The orphan diseases is like 5%. The 5% where it's just a small family or small groups of families, they've got some single genetic change, this is usually Mendelian, and they have a special disease all to their own. With whole genome sequencing, you don't have to hunt for these genes anymore. You just sequence the whole genome. You sequence all 20 of them in the family cohort, you run it through a whole bunch of computers, and then there it is. Nature paper. Wow. Right. There's a Canadian consortium, and Canada's leading this approach. We're early adopters of innovation, if we're not making them ourselves, called Forge. And in this, another group of Canadian or Winnipeg physicians, clinicians, people who look at genetic diseases, have grouped together in a consortium with all their colleagues across the country to come up with just the biggest list of every one of these kind of orphan diseases. Small families out in wherever, Manitoba, somewhere out in Quebec. And because the clinician is directly in contact with the patients, it's easy to get DNA samples. It's easy to get consent to look for these genes. And they just pour into the front of this machine of new sequencers, and out pops, here it is. Here's the gene that causes this family's disease. Here's the single base pair out of those 6 billion that drives it. Right? Unbelievable. 5 years ago, impossible to even imagine. And this project in 18 months will sequence and identify 150 rare diseases in Canada. Incredible. 18 months. You can barely get a project started these days in 18 months. And they're going to do 150. I don't know how many issues of Nature this would be, but I'd like to be publishing 3 or 4 Nature papers a month, I can tell you that. All right. So it's here, it's happening. It's changing our regular way of doing medicine. So don't worry about these slides. I just throw them up there so you can say you saw some complicated slides. But as you all know, you've got 2 copies of every gene. You've got 2 copies of every chromosome. And we call this a haplotype. Your copy of one gene is the one your dad has. The other copy is the one your mom has. The only copy your mom has is the one all her whole family has, et cetera, et cetera. So when you look at genomes in a kind of larger viewpoint, it's really a much smaller number of haploids or sets of kinds of chromosomes. And this is a way we've gene hunted before. Try to find a whole arm of a chromosome that's consistent with the gene. Then you jump in and you try to use markers to see what just changed, who's got the disease, do they always have this version, et cetera. With whole genome, you can not just try to find these in-your-face kinds of changes, you can go to the single base pair changes. Those one base in 6 billion that's different in the patients that have the disease and that seems to always be associated with that gene. This is called SNPs. You don't have to remember that, but it just means it's single base pair changes that somehow change the expression of that protein. I like to call them just flavors. You've got one gene that does something, and remember, we don't really know what they all do, but in this room, there's probably 20, 30, 40, 100 flavors of that gene. It's the same gene, almost the same sequence, just very small changes. Sometimes those small changes have a really big impact, and when they do, you have a heritable genetic disease. Being able to look at, whole genomes means that it's pretty straightforward now. It's not easy as pie. I don't want to give you the wrong impression. But you can see here, you can look here and you can say, hey, what's in the red ones that's not in the blue ones? Out of 6 billion, you've found 5. They're in the same gene. This is now starting to be pretty straightforward in the hands of people that can do this. So how about complex diseases? So here we're looking at diabetes and Crohn's disease. Crohn's disease is an intestinal blockage, kind of very painful lifetime problem. Diabetes, also not very good, lifetime problem. And quite often people who have one develop the other. And so we know genetically there's some link between these. And we know lots about this, right? We aren't just blindly figuring things out now. But and just look at the different colors here. They're related things about immunity, about way cells interact, about way cells die, the way cells proliferate. And you can start to put things in bins. So you can say, well, there's probably at least one gene in each of these bins that together is going to tilt you a little more towards getting this disease. And we know that if you got this one, some of those are already going to be pushing down the other one. And then we try to figure that out. That's kind of current genetics. With this high throughput thing, there's no more guessing. You can do the whole genomes. You don't even have to know where the genes are, right? You just have to do the whole genomes, then you know where all the changes are. And it starts to change the way a clinician might look at this disease. Right? There are 28 loci in common. There are some unique to each. And so you can start, if you know all this information, you can start to say, well, if you got this disease, you got a pretty good chance you're going to get the other one, right? Or maybe you got really unlikely chance you're going to get the other one. These are this is totally new to medicine, to be able to kind of make statements like that. And in fact, it's not just the genes really, right? It's the biology of the genes that drives it, and you can start to figure that out. Suddenly, these big Venn diagrams become this very simple one. There's 2 flavors, 2 groups of each in terms of genomes that are more likely to get one over the other, and one kind of that over the other kind, right? So this is starting to change diagnosis. It certainly is the preliminary data to say it can be investigated further to actually make diagnostic decisions. Breast cancer is another great example. Breast cancer is always clinically diagnosed by pathologists, right? There are 11 kinds of breast cancer, and they almost always fit or look like one of these. And it's a pathologist that tells you which one you have, confirms it, and it's the clinician that then decides your treatment. Right now, breast cancer is quite a bit along than other diseases. We already know if you have estrogen receptor, you're going to be in a little bit better situation than if you've lost the estrogen receptor. Already binned. This is genomics, right? Genetics, really. So if you're in this group, you get this treatment. If you're in that group, you get a different treatment. That is personalized medicine, right? That's a clinical decision based on your genetics of this particular receptor. When you start doing this new approach and you say, oh, let's sequence a whole bunch of cancers and then do cluster alignment, and there's so much bioinformatics. If you're thinking of going into computer science, we need you, right? This is where the data mining all comes in. And so there are many different kinds of breast cancer, many more than 11, if you look at the kinds of genes that are expressed or the kinds of changes in genes that are individually different between groups of people, right? Not individuals. Collectively, we're looking at changes in groups of people. The billion ways, this is by no means over, although I think now this alone has changed it to somewhere in the 20 to 30 range of kinds of breast cancer that are clinically diagnosed. But you can play with it, right? There's a lot of room to go. You can see here by these colors, you can run an algorithm, and they'll all come out the way they should come out if you hadn't done this. But if you start looking at clustering them based on other things, the biology that's associated, for example, or whether, and this is another new thing, whether the drugs that are used to treat this will have any benefit to you, you start to get very different clusters, right? So now it's not just diagnosing the disease, it's tying that to the treatment you're going to get and whether that treatment will actually be any good to you. A lot of chemotherapy in cancer makes you very, very sick. If you already know that the genes that normally metabolize that are absent in you and the whole thing's not going to work, there's no point in getting really, really sick because that medicine is not going to help you. Likewise, you can take a lot less of it if those enzymes are really active. And so this is personalized medicine. This is using the flavors of your genes, looking at the entire genome, to determine both the diagnosis of your disease and the effective treatment of your disease. Personal genomics. It doesn't mean you got a green jacket, I look at you all. It doesn't mean it's custom tailored for your genome. It's what we know about genomes and your grouping with others and the science and medicine behind that that groups it into personalized medicine. Okay, so this approach, Mendelian traits, plowing through this. This might be done in the next couple of years. It's changing, allowing us to go after the really big killers. Right? This next 10 years in medicine is going to be very exciting. It's changing today the way diseases are diagnosed. It's changing today the treatments. Low-hanging fruit, I'll give you that. Small steps, though. All right. So I'm often accused of making things too exciting. I don't know if you're excited. I am, obviously. But here's some cautions. Okay. The more we know, the more we don't know. So this genome thing, it turns out, isn't just genes. The genes are only like 15% of your entire genome. We're finding out something called epigenetics. You may have heard of this. This is non-genetic changes in your chromatin, in the whole package of your DNA code that really change gene expression. Okay? So you've got one fertilized egg. It's very different than that pluripotent stem cell that I use in the lab that can still make all cells in the body. This is certainly very different than the 200 universally different kinds of cells that you can have after. They all have the same genome. It's epigenetic regulation that controls the program or suites of genes that are expressed. So this is meant to say the code isn't the whole story, right? It's how we regulate that expression of that code. And so hope. This is also new approaches to treating diseases, and as well as you're not doomed because of your genome, right? Many ways to change it, even genetically. So, second thing, billions and billions of four-letter codes flying off of these machines. How can you can't see it. A gene, I can draw out my favorite gene and I can tell you a lot about it. I can see it, I can hold it, I can taste it some nights. Genomes, no way. So there's a lot of new areas coming in. How do we visualize this data? How do we see it? How do we, put colors on it so our brain can recognize patterns? We can make these innovative leaps. There's a whole new fields here for people in computational analysis, people in bioinformatics, people in design, architecture. These are going to be the giant breakthroughs. We've done the code. It's how do you make sense of this for everyone to see? That's another big challenge. So I want to leave you with some concluding, hopefully upbeat points. There's a cover of Time when epigenetics was first discovered. Your DNA, your genome, your code, even though it's on your health card, it's not your destiny, right? There's a million. I just talked about epigenetics. There's all kinds of other things in there. Non-coding RNA genes, microRNAs, chromatin remodeling, gene deserts. Half the genome we don't even know what's in there. We actually haven't sequenced it. So there's a lot to learn. But one thing we know for sure, these genetic or genomic things which you can actually say and show and point to, these small changes, it's a risk. It's not destiny, right? It's a piece of information with some kind of best guess probability next to it, all right? And genetics is really exciting, but genetics is nothing without the environment. I'm sure you've all heard that, genes versus environment. Genes are designed to play off the environment because that's where we live, right? And so genes aren't the whole thing, and genomes certainly aren't the whole thing. You can make changes to your environment. You can make changes to your nutrition. And you can certainly make changes to your lifestyle. you have a lot of control yourself on the outcomes of your genomes and how they may or may not be expressed. All right. So the last word, well, we already answered this. I gave it away in the first slide. Yes, it will be on your card. Maybe not me, maybe you. Certainly your children will be the first to have it done at birth. Right? That's the most likely scenario. Which will be cool, eh? Because you get to hold that card for the first 18 years. So that'll be fun. So I want to leave you with this. This is a crystal ball. It's really exciting. Genomics and genome personalized medicine is not future telling, right? It's horoscopes. My wife reads horoscopes every morning. My mother-in-law does. And whenever something really important, oh, hey, guess what happened? Right? It's just like that. The horoscopes do not have any meaning without your own context of your own life and your own lifestyle. In the same way, the genomes is not an absolute destiny, fate mapped outcome for you. It's in the context of your own life, your own situations, and you have a lot of control over changing that before you might ever get to that point of disease. So I'll finish there, and I'm happy to take any questions. What is the difference between genetics and genomics? Genetics is looking one gene at a time. Genomics is looking at all 22,000 genes at the same time, plus all those other bits we just talked about at the end. So it's a different way of looking. It's not this tool will do that. It's here's the whole problem. How do I use that information to attack it? Does that answer your question? Yeah. Yes. Sorry. You, and then you. The question is epigenetics and chromatin remodeling happening in stem cell research? Yes, that is kind of the low-hanging fruit, the first kind of discipline in current sciences where it's really tractable. So stem cell biology looks at cells, progenitor cells that have a lot of capacity to become many different kinds of cells. And what we're realizing as we try to harness that is that it's not just genetics. We know transcription factors, we know pathways, we know things that are important to make a neuron cell. But it's the epigenetic remodeling of gene expressions that drives it to be a neuronal cell. So it's just the start. It will be important in many other diseases. And I can give you some examples. If you know a certain pathway is really impacted genetically or genomically in diabetes, for example, you know the whole story. You might know other pathways to get around and repair that function. You can do that epigenetically by turning on those pathways without changing the genes. In the purple, you had a question?
Why is it so beneficial for everybody to have a genome on their health card? Because, I mean, I understand the value of the information, but on a health card, I mean, we're looking at so many things, right? So why do we have to have it so...
So the question is, why have your whole genome on your health card when, you know, you can't read a chip? It's quite simple. It's all economics. This is the $1,000 genome, and economics we talked about today too. You know, a single genetic test, which we're not very good at doing yet, even though we know a lot of gene-by-gene diagnosis, probably costs $5,000–6,000. So you can do one test or get the whole thing and never have to do a test again. That's the real reason. Okay, you had a question here?
I see this pattern that technology is ahead of us. So I see this great technology that we can have this information. My question is, how do we absorb the knowledge? For example, diagnosis, for example, because you have the 20, there are thousands of, so basically you're more precise at locating and identifying that gene. But the thing is, number one is how to process information with existing
Okay, so the question was, this is really exciting technology, but it's surely got to be overwhelming. How do we process it? How do we deal with it? Okay, so I'm speaking from a medicine point of view. It's science, engineering, technology that is going to drive, that is driving the technology we use, and is the answer to your question. So I just, a bit of a chicken and egg question. Technology always seems to be ahead, but I just want you to consider the reason that that technology was developed was to solve the problem that's really based in medicine. And just every now and then you're really good at it, and now all of a sudden you have too much information, and you realize, oops, the next problem is how do I see all of that? I gave you an example for that. And you know, this SNPs, these flavors of genes that I was telling you about, between you and the person sitting next to you, there's probably 100,000 differences between you and even your brother, right? So that's a ton of information. Yet already we can do this. If you isolate a gene and start looking at it and at that level, you can start to make sense of it. So it will follow.
I think my point is, I think the medical field should work with the business field so that we can better communicate the knowledge and also better utilize it in a more applied sense.
Absolutely.
That's really what I
Absolutely.
think the gap is. That the technology is way there, and we're not saying it's bad. It's just I see that lab, that the gap in terms of the application.
So in science we call this multidisciplinary, okay? So I'm multidisciplinary because I talk to the guy or gal in the lab next to me, so I get to make this big claim. That's not good enough, and you're absolutely right. It doesn't count if I talk to another one of my faculty. I got to come down here and talk to some engineers, to some marketing experts, to computer science folks. And I can tell you, we all do a heck of a lot of that right now. And the way funding is going forward, we can't move forward without it anymore, right? Gone are the days where you're in a lab and you can close the door and not come out till 8 when you go home, right? Everything now is transdisciplinary. The funding is transdisciplinary. Has to be. It has to be. A geneticist, I can't solve all these problems. I sure know how to use them when they come. So like as you were saying forward, before, yeah, I watch everything, read broadly, and that's where big leaps come. Yes?
Are there any legal barriers to human gene therapy right now?
Oh, lawyers wreck everything. Yes. There are tons of barriers. So you can't have a business if you don't own it, right? This is a long-standing problem. But scientists can't wait and don't wait. So we're under this illusion that as an academic professor, I'm somehow removed from these laws, and I just go ahead and do it anyways. Because if I find something good, people won't really care, right? That's kind of been the recent practice. The more modern practice is actually governments are actually making that law, right? All of my research is funded by your parents and some of you in the room. Why shouldn't it be available to anyone who can use it without licensure and without, I'm going to get in trouble for saying that out loud, so don't tell them on your next meeting. But this is so-called open access science. Everything I do goes from my computer straight onto the internet. All these sequencing that we're talking about goes straight from those computers onto the internet. Right? It's making as much information and technology as we can, reasonably, available to anyone who wants to use it or can use it or has the next great idea. That's not to say I don't believe in licensing or patenting. I think it's a balance, right? If you really want to take something to market, I find the cure for cancer and suddenly I can't sell it because nobody wants to make it, that would be a sad story, right? So it's a balance between the two. All right. Well, thank you very much.